Good day and welcome to this re-recording of my talk on AI enhanced knowledge mining with KnetMiner. I'm Arne de Klerk. I'm a co-founder and the product manager in the KnetMiner team. I've always had a big love for Kew Gardens ever since the first time I visited, and so I was really fortunate to have followed on from Paul Kersey's keynote at ISMB 2025. It really got me thinking about the beauty that can come from collaboration, because Kew Gardens is such a long-running success story, which was fundamentally built on the research from across the world. Kew also just so happens to be one of my favorite places on Earth, and my wife and I found our favorite trees and roses there, and we even grew some strawberry hill, which we first found at Kew, from root over the last year. Inspiration really is the driving force behind almost every great thing in existence today. Much like how Kew inspired us to grow our own garden at home, I believe that the driving force behind many plant scientists today is to feed the changing world. From ancient crops to today's complex supply chains, agriculture has always been about survival and vision. But our generation is now facing a bit more of a unique urgency. Climate change, shifting diets, and resource loss all means that we have to double our yields on the same, or in some cases even shrinking land, with less water and in less time. If you forecast based on the current birth rates, it's expected that there will be 10 billion people on Earth by 2050. What it means is that science can no longer afford to be slow. As of today, thousands upon thousands of discoveries have been made, and the solutions to many of our problems are often just kind of out there, but they're buried underneath variations in schemas, formats, stacks of papers, databases, genome varieties. There really isn't a single scientist alive today who can connect all of the dots in plant science manually. And honestly, if you ask a plant scientist working in the industry today, they'll tell you how it can take them weeks or months to map out the most key pieces to even start their research. And even that will just be within one or maybe two species. That's where NetMiner comes in. It helps researchers to rapidly discover meaningful gene-to-evidence relationships, generate new hypotheses or confirm existing ones, and accelerate crop improvement by better understanding the current landscape around your question. It brings together data like an inspirational cloud to fuel your new or ongoing project. The tool has been developed over the past 10 years, and quite recently we've spun it out from Rothamsted Research, which is the oldest agricultural research institute in the world. Our goal is to accelerate the search for plant science knowledge and intellectual property, especially in the fields like crop improvement and food security. The app combines bioinformatics, knowledge graphs, and AI, and NetMiner connects fragmented data and unveils connections between genes, traits, and diseases that more traditional, more manual searches are likely to miss. There are over 1,000 plant science organizations and 200,000 plant scientists worldwide in academia and industry that can benefit from a tool like NetMiner. Wouldn't it be nice to know by day one of starting a new research project that you're not going to be repeating work? Wouldn't it be nice to have a quick summary, a fast and a vast understanding around your precise questions, all within just a few minutes? With manual searching, you're always feeling like, I'm just missing something. It's because scientific discovery, especially in genetics and biology, can often feel like you're sifting through an endless sea of information. Researchers will commonly face three main challenges: fragmented data, complexity of traits and diseases, and data overload. NetMiner aims to solve all of these issues by using knowledge graphs together with AI to make researching existing data more accessible. I want to talk to you today about a bit of a use case which will use the new GWAS peak search feature in NetMiner. Under Rothamsted, as part of the Delivering Sustainable Wheat program, we work with different wheat populations in the field, and we measure different traits. This phenotypic data can then be used and correlated with genotypic information, which is called genome-wide association studies. In the photo here, you can see a nested association mapping panel, or a NAM panel, which used 25 Watkins land races grown at Rothamsted. The population was grown and phenotyped and at the same time genotyped. That information was then used to run statistical analyses to find regions in the genome correlating with specific phenotypes. What you're looking at here then is a Manhattan plot relating to a GWAS analysis done for 1,000 grain weight. 1,000 grain weight being quite literally the weight in grams of 1,000 grains of a crop. It's typically measured to assess the grain size and the quality, and it's usually used in cereal crops like wheat, barley, maybe rice, as a key trait, since it's directly linked to the potential for yield. Here along the x-axis, you can see chromosomes and the locations of each of the 35,000 SNPs. The y-axis is then showing the significance per SNP. SNPs above the significant threshold are called quantitative trait nucleotides, and they mark a very specific position on the genome. I'm not going to go into detail about the statistics that are involved here. Instead, what I want to talk today about are the steps that follow down from this analysis. Because you may be wondering, what are these dots representing? Are they in specific genes? If so, in which genes? And how do we begin to interpret the data? How wide beside the SNP should we even look for those genes? Of course, the challenge is that those regions can actually contain dozens of genes, which could have limited or maybe some really generic annotations. So the question then becomes, how do we prioritize what's worth looking at? What most researchers will do at this point is now go into Ensemble, look for the genomic region around the SNP, and then start pulling out gene IDs. You'll get a list a bit like this. So you've got dozens of genes. Some have vague or generic annotations, but there's definitely no ranking. There's no context, and there's no indication of which ones are actually relevant to our question, which remember was which genes are related to 1,000 grain weight. This is now a really shallow search. It gives you what's there, but it doesn't tell you what's important or applicable to what you're looking for to begin with. Deep research, on the other hand, requires a lot more searching the literature from abstracts to patent data, searching published GWAS studies, annotation data like Go and TO terms, interacting genes, and more specific data like co-expression and enrichment data. You might also find that there is limited information in your species of interest, and so you may then have to stop and look at searching for orthologs and maybe some other species, and then start to map that evidence back. Or you could just try using a knowledge graph. By structuring the data using ontologies and schemas, property graphs can take away a lot of that complexity. Knowledge graphs contain nodes with edges between them. Both nodes and edges can then have properties which will add additional context and meaning to the graph. If you pair knowledge graphs with a tool like KnetMiner, which lets you search, filter, research, visualize, and summarize the results, you're on the fast track to discovery. So let's take a quick look at what that would look like. Here I'm in NetMiner, I'm in wheat, and I'm just going to run a GWAS peak search. So I'm uploading a file here, some GAPIT results. I've uploaded that file, just making sure the heading is going to map correctly, importing the data, and I can see 6 of the 35,000 SNPs were found to be significant on the bond for any correction. I'm just changing the SNP window region to try and capture more genes in those regions, and here we go. So we've got 6 genes, and we can see all of the evidence relating back or relating to those genes inside of the NetMiner knowledge graph. Now I'm just taking a look at some of the filters inside of evidence view, all the different evidence related to those genes, and I'll just jump back into gene view, open up the filters again, and what I'm going to do here is just add a keyword filter for grain, because remember we're looking for 1,000 grain weight. Now you can see it's cut us back down to 3 genes. I noticed 2 of them were actually on chromosome 4A, so I'll just filter for those, select the 2 of them, and use them to create a network. And here we go, here's that network. I'm turning off the node grouping, and I'm going into the Graph Explorer and adding in some cultivar information. I'm just going to relay out the graph a little bit here, so we can see a bit more clearly what's going on. And there we can see our two C genes, these 2 here with the double blue border, and how they've got all these blue edges which means that they're actually present in all of the different cultivars that we've got available inside of NetMiner, which is quite interesting. I'll now jump to the next video. Here I'm going to now add some more publication information into the graph. And I can see some of them were connected into the current graph. A few ended up floating around, so I'll just remove the disconnected ones. Now I'll jump into the AI summary feature, which is using a RAG-based approach. And so here I've had the gene list pre-populated, and I've put in TGW, 1000 grain weight, and grain as my two focus topics. I'm telling NetMiner I only want you to use the visible publications to create this summary, assuming of course that I've already had a look at what's in the graph and chosen maybe more recent or relevant publications for my summarization. And here we go. Here's the result. I've got a quick way of even linking back into the graph. As you notice, I clicked on an in-text citation there, and that opened it up, selected it in the graph, and I could see the properties of that publication and even click through to an outlink, which took me into PubMed. So just jumping back to what that literature review looked like, here it is, just a bit bigger. And because of how the language model is being prompted, it's always got quite a consistent structure with a simple overview at the top, a review then in the middle, and at the bottom it's got a trait summary and citations section. Like I mentioned before, hyperlinks are throughout the text, and it always allows linking back into the graph. It's really great for then understanding the publication and its findings in a bit more of a connected manner. I'd like to wrap up this use case by showing how we've narrowed down our deep research. The wheat genome has around 120,000 genes. Our GWAS analysis contained 35,144 SNPs, of which NetMiner identified 6 to be statistically significant under Bonferroni correction. Combining that with some more downstream filtering and searching, we were left with 2 strong candidate genes, which can now move forward for experimental validation, maybe some gene editing or transformation, and then their effects on 1000 grain weight could be confirmed. Of course, as with everything in every software product, there are many challenges, and I'll just be talking about 2 today. The first being in data integration. We integrate the same biological and genomic data sources that you would otherwise manually scour during your research. Our ETL pipeline combines data from various sources like Ensemble Genome, Ensemble Plants, Fungi, and Compara data, to PubMed for publications, and then more specialized data sources like TARE, UniProt, and Delivering Sustainable Wheat's own wheat data. Our ETL pipeline processes and then unifies these data sets, and collections of species are then made up into Neo4j databases in our backend, and then we point a NetMiner API to that Neo4j instance to make that resource available to the front end to use. Here are 2 lists of identified candidate genes. Both of the studies are valid ways of building gene lists, but they can really be noisy and maybe sometimes broad. So one is based on statistical linkage and the other one is based on functional expression, but neither alone is busy capturing the full story. That's exactly why we need more integrative approaches, like using knowledge graphs to combine this kind of data, and then that lets us reason across it. Now let's bring everything together. What you're looking at is a simplified view of how we structure biological evidence in the NetMiner knowledge graph. At the center of most questions, we've got a gene, and around that we connect various different evidence types. Differential expression data from transcriptomics, SNP trait association data from GWAS, disease and trait enrichment data, variant annotations, and each of these edges will have a weighting to them, like p-value, fold changes, effect sizes, and they're all listed as properties of the graph. But now, instead of treating this data as separate silos, we represent them in a single graph where relationships between the nodes are carrying meaning. Of course, a large amount of the biological insight is actually also buried in literature, hidden in abstracts, full text, supplementary table, patent databases, and this is where we layer on some text mining to further enrich the weaker or maybe more emerging signals. That then moves us into our second challenge, intelligent summarization using AI RAG. NetMiner uses retrieval augmented generation, which is an AI technique that combines information retrieval and normal language model text generation. It allows models to generate answers based on relevant external documents. In our case, those relevant external documents consist of the list of genes that were used to create the graph, the keywords that were used to create the graph, and the publications within the current graph along with their titles and abstract. The user has the choice about changing any of these parameters at the point of creating the literature review, and that just leaves a little bit more on the table for them to modify what they're expecting to see. They can also say if they want to just see the visible publications appear or all of the publications inside of that current graph. If you remember earlier, I showed the Graph Explorer and how you can add or remove any of the nodes from the graph. For RAG then to work well, the model actually needs context, right? But we need to keep in mind its limitations, its risk of hallucinations, and the problem with context window size constraints. What we've come up with to combat the context window problem is a solution where this little yellow region at the bottom is threshold based. So if more than 50 publications are selected for summarization by the user, we'll use a vector embedding database in our Neo4j to prioritize the publications that we want the model to consider based on how nearly they cluster together. Since we can ask the language model for a structured output as part of our request, we can also then build small features around that, like in-text citations, with the front end being able to recognize and allow the user to click and view the different publication citations in their network. That'll select a node, open up the info box, and they can read more about it. For me, this is really important, right? I've always felt that the best implementation for an AI is one that is tightly integrated with the underlying technologies and doesn't feel like either a separated or a bolted on solution. In moving forward with AI then, we've identified a bit of a roadmap for 2025. There have been huge advancements in on-model processing the last 6 months, with certain models now being able to actually do pre-processing and chunking of the data on an internal level. Since the model only needs to see a few relevant chunks and not the whole dataset, the model can then search the summaries it has created and provide itself with the most relevant context. These are actually the same kind of tricks that we have been doing ourselves to handle the problem of massive data during the summarization step. But now it's being more and more taken up by these advanced models directly in their product. It's very clever and something we are obviously looking into. Of course, this kind of work takes a mountain, and NetMiner is what it is today because of the fantastic work done by the team behind it. So a massive shout out has to go to all involved. Thank you so much for tuning in and listening to my talk. I hope I've inspired you to stay curious, to explore new research ideas, or to try NetMiner with your own genes or questions next time you're searching for answers. You can reach out to me via the email on screen, or come and have a look at our website on netminer.com and try one of our free resources today. Thank you.